The overall goal of this procedure is to use a room temperature surface passivation method in order to inhibit surface recombination, such that bulk silicon defects can be accurately characterized.
This method can help us understand what defects will limit the lifetime of high-purity silicon wafers, which are required for very high-efficiency solar cells. The main advantage of this technique is that it can be performed at room temperature, meaning the recombination activity of bulk silicon defects will not change during the measurement. The implications of this technique extend towards measuring the recombination activity of bulk silicon defects that exist in low concentrations, which otherwise would be very difficult to measure by other techniques. Generally, individuals new to this method will struggle because the requirement for high purity DI water and chemical cleaning procedures is not initially understood. Thus, it's imperative the procedure is followed step by step.
Solution preparation, including vital notes on hydrofluoric acid preparation, as well as general setup and equipment calibration instructions, are all provided in the text protocol. With those objectives achieved, proceed with cleaning the silicon wafers using chemical treatments. Begin with loading the samples into a quartz cradle. Then, transfer the cradle into a hydrofluoric acid bath meant for general use. After about 10 seconds, the samples will be hydrophobic. Remove them from the bath and rinse them off by passing them through 3 deionized water baths. Next, in a fume hood, slowly immerse the cradle in the SC1 solution, which has been maintained at a temperature